Welcome to today's lecture on the fascinating world of molecular biology and gene regulation. Today, we will be exploring the intricate mechanisms behind DNA structure, gene expression regulation, transcription factors, DNA-protein interactions, enhancer identification and classification, enhancer-promoter interactions, chromatin remodeling, cooperative binding and combinatorial regulation, transcription factor networks and gene regulatory networks, transcription factor mutations and diseases, transcription factor targeted therapies, epigenetic regulation of transcription factors, and experimental techniques in studying transcription factors and enhancers. So let's dive right in. At the core of gene expression regulation lies DNA, the blueprint of life. DNA is a double-stranded helix composed of nucleotides, which consist of a sugar-phosphate backbone and 4 nitrogenous bases, adenine, thymine, cytosine, and guanine. The specific sequence of these bases encodes the instructions for protein synthesis. Gene expression regulation involves the modulation of this process to ensure proper cellular function and development. Transcription factors are crucial players in gene regulation. They are proteins that bind to specific DNA sequences, called transcription factor binding sites, located in the promoter regions of genes. By binding to these sites, transcription factors either activate or repress gene expression. This intricate control allows cells to respond to various signals and adapt to their environment. DNA-protein interactions play a pivotal role in gene regulation. Transcription factors interact with both the DNA double helix and other proteins to exert their regulatory functions. These interactions can be mediated by specific protein domains, such as DNA binding domains and protein-protein interaction domains. Understanding these interactions provides insights into the mechanisms underlying gene expression control. Enhancers are key regulatory elements located in non-promoter regions of DNA. They can influence gene expression over long distances and are essential for precise spatiotemporal gene regulation. Identification and classification of enhancers are crucial to decipher their specific roles and understand the complex regulatory networks they participate in. Enhancers exert their regulatory effects by interacting with gene promoters. These interactions can occur through physical contact facilitated by DNA looping or through long-range chromatin interactions. Understanding the mechanisms of enhancer-promoter interactions provides insights into how genes are selectively activated or repressed. Chromatin remodeling refers to the dynamic modifications of chromatin structure to regulate gene expression. Chromatin, consisting of DNA wrapped around histone proteins, can adopt open or closed configurations. Remodeling complexes, including ATP-dependent remodelers, modify chromatin structure to facilitate or hinder transcription factor binding, thus impacting gene expression. Transcription factors often work in combination, and their interactions with DNA can be cooperative. Cooperative binding allows for fine-tuned and specific gene regulation. Combinatorial events involving multiple transcription factors binding to target DNA sequences enable the integration of multiple signaling pathways, resulting in highly complex gene regulatory networks. Transcription factor networks and gene regulatory networks involve intricate interactions between numerous transcription factors and their target genes. These networks coordinate gene expression programs in a precise manner to ensure proper cellular function and development. Disruptions in these networks can lead to various diseases and abnormalities. Mutations in transcription factor genes can lead to dysregulation of gene expression, resulting in diseases. These mutations can lead to loss or gain of function, altered DNA binding specificity, or modifications in protein-protein interactions. Understanding the impact of such mutations provides valuable insights into the pathogenesis of various diseases. Given the crucial role of transcription factors in gene regulation, targeting them has emerged as a potential therapeutic strategy. Modulating transcription factors activity can restore aberrant gene expression patterns in diseases and promote optimal cellular function. Transcription factor targeted therapies hold promise in the treatment of various diseases, including cancers and genetic disorders. Epigenetic modifications, such as DNA methylation and histone modifications, can influence the activity of transcription factors. These modifications can either enhance or restrict transcription factor binding to DNA, leading to altered gene expression patterns. Elucidating the interplay between epigenetic modifications and transcription factors provides insights into the complex regulation of gene expression. A variety of experimental techniques are employed to study transcription factors and enhancers. These include chromatin immunoprecipitation, CHIP, CHIP-seq, electrophoretic mobility shift assays, EMSA, and reporter gene assays, among others. These techniques enable the identification of transcription factor binding sites, characterization of enhancer activity, and investigation of transcription factor function. In conclusion, Understanding the molecular mechanisms underlying gene regulation is crucial for unraveling the complexity of cellular processes and diseases. From DNA structure and gene expression regulation to the experimental techniques utilized in its study, the intricate interplay between transcription factors, enhancers, and chromatin provides a foundation for targeted therapies and a deeper comprehension of human health and disease.